So as Marcus said, I am an ecologist and a scientific illustrator, and today I'm going to be talking about the art of science, how we bring those two worlds together in ways that sometimes people don't even think about. And it's going to be a little bit of my story and my process on how I think about these things and bring that creative um, thought process to the scientific work that I do. So I'm going to go out on a limb here and say you are here present today, either on Zoom or here in person in Henderson or up in Reno, because you are curious. You're curious about a lot of things, probably about the desert, about the Mojave, about the Great Basin, about plants, animals, these systems that all work together, but other aspects of your life, music, um, entertainment, books, literature. You're curious. You like to explore and discover new things.
Yeah.
My curiosity began at a very young age. My parents didn't have a lot of money, so they pretty much, my mom would take us to any free museum that she could find, free library events that they would host. And so that love for both the sciences and arts started at a very young age. And I was very fortunate to have parents that encouraged both of those loves, both illustration, drawing, gave me paints, crayons, and then also that scientific curiosity as well. But as I grew older, there was kind of a fight that started to happen between science and art and which one I would choose kind of in my own brain and just the feedback I was getting kind of from the world. You'd hear like, oh, you don't want to be a starving artist, or like you wouldn't want to waste your money on like an art degree. I actually, when I was in high school, wanted to be a Disney animator. I was like, yeah, I'm gonna go animate some things, and then Disney shut down their animation studios. So that was that. But at the same time in high school, I had an AP Environmental Studies class. And I was like, oh, environmental justice issues, the intersection of science and people, and how this changing environment can disproportionately affect people. And so that opened a whole new world to me. And so when I entered USC, I actually got a BA in environmental studies in environmental justice issues. I thought I wanted to go down that route. Um, and I asked the art school, can I double major in fine arts and environmental studies? I was told no to my face by the admissions officer for the Ross School of Fine Arts, that they do not like their fine arts majors double majoring in the sciences because they know that I would put more time, or their students would put more time into their labs than their artwork. And I just remember, it's a core memory. It's one that I can close my eyes and I can pull up the room, I can pull up the light, I can pull up exactly how things are placed, and her sitting there telling me that. And I think a lot about that, like when, especially for teachers, people who deal with technicians or young people beginning in their fields, like things you say to them that could become some of those core memories that they pull back. And I remember also thinking, nah, I'm not going to do that. So what I did instead, like, you don't know me, you don't know who I am. And so what I did instead was a two-dimensional studies minor, which translates to I took whatever art classes I wanted. So I took um, printmaking, which was instrumental. I took painting, drawing, and I made it a point to include art into the work that I was doing in my major as well. So I was determined, and so I did a project, a capstone project, my senior year on the Los Angeles River. LA has a river. It is a navigable river. A bunch of people kayak down it, and it is a navigable river. It pumps out as much water as the Mississippi in peak flood events. It's phenomenal. and there's a huge revitalization effort that's going on. So I wanted in my capstone project and for my major to investigate the LA River from environmental justice standpoint, but also from ecological standpoint, and additionally from an art standpoint, and use that to combine forces. So this piece, it's a 4 by 6 foot unprimed canvas, and I actually went to the La Brea tar pits in Los Angeles, where there's naturally seeping tar that comes up from the earth, and I'm sure there's a security camera footage of this somewhere that exists, but my friend drove me up. I jumped out of the car, ran up with a cup, grabbed some tar from a place where it was bubbling up outside the fence, hopped back in the car, and we drove away. And I used that with a little paint, like thinners, to paint on the unprimed canvas. So I was particularly interested in kind of the chemical process of this paint, of this tar kind of eating away at the unprimed canvas over time. This thing took months to dry. It's now on my wall. It's finally dry, but it took months to dry. And so I was interested in that intersection between processes on the canvas that are, there's a temporality to it, where these pieces may not last forever. Just like if we don't take care of our systems, in this case, the LA River, those don't last forever either. I also, as part of this, did anthotype. So this is a printing process where photography process, one of the first ones actually, where you can actually develop images on the extract of fruit. So the larger image is an anthotype that's developed on beets or beetroot extract, and the smaller one is developed from strawberries. I found that strawberries worked really well. So this process, you take the fruits, you crush them up, you put it through a cheesecloth, you get that nice pigment, you coat paper with it, you put a either a negative or a positive, in this case, a positive down, and you put it in a light box and you set it in the sun. And so I also described the chemical process that's happening. The light is bleaching through to the pigment, and after a few days, you're left with the image where the sun did not bleach. Here's another strawberry anthotype. And I did go out and take these pictures of the LA River over a course of a few days myself. And then I also did chlorophyll printing. So this is a process where you can develop images, also an early process where you can develop images on leaves. I only had a few of these that turned out well, very tricky process to do. I actually moved it because I didn't think it was working, and I moved it, and then I saw it and I was like, oh, it's working. So I tried to put it back, and then it was too late. But it's a process of developing those images. So similarly, light bleaching through in a light box, but you kind of have to keep the leaf alive while the process is happening so it doesn't dry out. And again, all of these pieces have that temporality mixed in, where if it's taken out and then exposed to sunlight and light, it's eventually going to fade. So unfortunately, none of the anthotypes or the chlorophyll prints exist today. And I wanted that to be a part of the expression of the environmental justice concerns and the ecological concerns surrounding the LA River. There are ways to preserve them that involve resin, sealing them in resin, but that was just out of my scope at the time. But I decided I would submit to the USC Undergraduate Research Symposium in 2011, that whole body of work. And I actually won second place in my category for this intersection of science and art. And I sent that picture of me to the admissions officer, who was still at the Roski School of Fine Art. And I had the full support of both my advisors, my printmaking professor, and my advisor in the environmental sciences department. And sent it to her, and I just said, hey, look what I did. And you know what? I didn't get a response back, but you know what? I never looked back. Since then, in my master's and at the work that I do at the Desert Research Institute, I have found a way to combine science and art in those creative processes. And I'm going to go through a little bit of that today. So scientific illustration is not a new subject by any means. Back in the day, scientists didn't have fancy graphing software and computers to document their findings. So they relied on artists, or in many cases were artists themselves, to document those findings and preserve them to spread that knowledge and research to other researchers in their fields. So scientific discoveries at this time were actually propelled and preserved by artwork. And there's, of course, really famous examples of researchers, naturalists who were artists themselves. So these are Darwin's finches, he drew them, and of course, Audubon's birds. So this was the golden age of scientific illustration, where it was not just other researchers that were gaining access to these illustrations. It was a way for the members of the public to also engage with the work that was being done, go to these faraway places that they had no ability or access to ever get to. And it's important. For example, this is a picture or an illustration of the Tasmanian tiger. There are photographs of these species before it went extinct, but they're only in black and white. And so these illustrations are really the only color representation that we have of them before they went extinct in 1936. But do we put ourselves in silos? You hear it a lot from scientists who are just like, I'm not creative, or artists who are like, I can't do math. And there's a lot of like compartmentalizing that can happen, especially when you're going to college and they're like, pick a major, and this is what you're going to do for the rest of your life. And you're like, oh my God, I'm 18. What is going on? But that's where art can come in when it comes to scientific communication. I really like this this tagline. So scientists, stop thinking, explaining science will fix things. And this was actually published in 2017, a few years before COVID, where we saw that play out in real time in a very, very unfortunate way. Art can actually be, as Marcus said in the intro, art can be a very powerful conveyor of scientific fact. We need to take our research out of our publications, out of our manuscripts, and put it in an accessible form that can be spread to a broader audience. And what better way to do that than one of the universal forms of communication, which is artwork. And I'm talking about fine arts here, so like painting, illustration, but art comes in various forms: pottery, music, so many different forms of art and creativity. So there is that rich history of artists and scientists collaborating together to bring data to life. So at DRI, one of the things I have done in the past is working with researchers at DRI, but also around the world to bring their data to life. So this is a graphical abstract that I did for some researchers out of Perth, Australia, where they were wanting to describe the effects of rodenticide moving through the food chain in Australia, and then also the broader implications of that research around the world. And they had no idea that they needed a graphical abstract for their publication submission. It was required by the journal, and happened to see my work on a different author's paper and reached out. And I'm always happy to help bring that data to life. And it's a collaborative process. They have the idea, And sometimes I'll be like, all right, sketch it for me. I don't care if I get a stick figure frog, give me the stick figure frog. Um, and then we go back and forth. I'll do a first iteration, they'll give me comments. It truly is a collaborative process. Um, and oftentimes I'll show them the final result compared to what the original was. And they're astounded by how close it is. I'm like, you can be creative. Science also is inherently a creative process of designing an experiment, visualizing the data in graphical form, troubleshooting when problems arise in the field, laying out the framework of how plots are going to be set up. So I tell scientists all the time, don't discount that creativity. So I'm going to go through a few just examples of projects at DRI where I have combined that art into what I do. The first is through a botanical inventory that we're doing at Tule Springs Fossil Beds National Monument. I can't see people online or in Reno, but for those here, has everyone been to Tule Springs up in North Vegas? Yeah, it's great. It truly is an urban park. It butts right up to people's houses. There are now going to be houses developed almost in the middle of it. It's wild, but there are beautiful resources out there. It's designated for its paleontological resources in the Upper Las Vegas Wash, but it's home to a lot of animals and plants as well. So I know you guys probably all know this, but when I talk to people from other parts of the country or out of state, they're just like, the desert. I've been driving from like Reno to Vegas and I've seen the same shrub 50,000 times and it's brown. And I'm like, okay, all right, hold on. It's a very special place that we live in. I describe the desert as stark, subtle, and sublime. It favors those that take the time to get to know it a little bit better and look a little closer. So Nevada is 11th among all states in total species diversity, 6th among all states in the number of unique or endemic species. That includes 64 recently discovered endemic species of spring sails, 8th among all states in butterfly diversity, and 9th among all states in mammal diversity, and our plant diversity is quite high as well. This is from the Heritage program. I'm sure I should ask them for an update on it, but we live in a remarkable state. And when I tell people to think about creativity and using those creative, I call them other lenses to view the world, where it's moving from the valley floor, creosote, burst sage, going up to Mount Charleston and watching the vegetation communities change around you, having the Joshua trees and then the juniper and then the pinyon and then bristlecone pine all the way at the top. It's not taking things at face value. So having a little creativity in the way we think about our systems as well. So I'm out collecting at Tule Springs. It's an ongoing process. I could ask Jim Holland. He's been collecting the Newberries for decades, and the checklist isn't done. So it's definitely a lifer project for me to go out to Tule Springs and see what's out there. I pride myself on collecting cacti. I like doing it. It's a little masochistic, but I feel like I've done quite well with some of the specimens I've preserved. And if Jerry Teen's happy, then I'm happy. So yeah, so it's fun. It involves filleting in the field and drying out things. I stopped taking them outside to swap the sheets because I was like, it's cold. And so I haven't got a choice finding my foot in my room yet. So here we are. But as part of this checklist for Tule Springs, which could become a flora if I'm feeling spunky enough, I would like it to be illustrated. And so I'm started to illustrate some of these for the Park Service. So this is Las Vegas buckwheat. glorious plant. I'm hoping to do a field trip, hopefully next year in the fall, to see it in flower. And then, of course, we have Las Vegas bear poppy and white bear poppy, both. So this park has designation of having 2 out of the 3 Artemicon species present within the boundaries. Unfortunately, one of them is still not present above ground. So it hasn't been seen above ground for several years now. We just haven't had the rainfall for that seed bank to respond yet, which is very unfortunate. And I collected it. It's formally documented now. And within a year, it no longer exists above ground. So it shows you the importance of these voucher specimen collections. Of course, collected on a permit. I have been asked that before by someone who was like, "You collected the last one?" I'm like, "No, no, 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 no." That's not how it works. It wasn't like there was one there and I took it. So yeah, permits and all of the above. But yeah, we're waiting on that to come back. The park also has a nice Phacelia diversity. So we have the 5 species we found so far illustrated here. And again, when it comes to scientific illustration, it's a great example of similarities and differences within the genera. And while you could look at a drawing and be like, all right, this is Tiffany's style, it still has to be accurate. Also at Tule Springs, we ended up doing a bear poppy root excavation. This bear poppy was already deceased, and it was an area that was within park boundaries, but in a right-of-way. Long, complicated story, but we were able to kind of get something out of the construction that was about to happen there. And it helps to dig a soil pit when you have an excavator. And I called my friend up in Reno, who's a geomorphologist, and was like, "Hey, want to dig a soil pit with me?" And he's like, "You had me at soil pit." So he came down, documented the soil columns, and I drew it to scale the root in the field in the ditch. And it was a 4-foot taproot. And we did not definitively find the bottom. It got down to about a millimeter in diameter in points and widened back up, which I've graphically kind of demonstrated here on the right, and just absolutely phenomenal. And my coworker was actually like, "Oh, you should design this figure. Like, I'm not really sure how to do it. I'm not creative." And he was actually the one who came up with kind of putting the lines and all of that. And I was like, there you go. That's the creative process. Now you can't say you're not creative, so stop saying that. But yeah, pretty remarkable taproot, which explains a lot of things about why it doesn't like being transplanted. Yeah. All right. And also, this was a project that I started at UNLV in the Abella Lab, where I was doing a seed bank project, and I wanted to create a unique guide from hobby plant species because I was getting really frustrated at trying to figure out what seeds were coming out of my extraction samples. And I was like, where's my seed guide? And then also growing out samples in the emergence assays as well. So I started taking pictures of the little seedlings as they came up. So yeah, identification tool for Mojave Desert seeds and young seedlings. That's a bear poppy seed. And the large plant. So this guide, it was the 3rd chapter of my master's thesis, where I did illustrations of seedlings using traditional scientific illustration process. And then I also took pictures of seed using stacking software with a DSLR camera mounted to a microscope. So a more contemporary scientific illustration technique. At DRI, I was able to take this project, started with my master's, apply for funding, and BLM California generously supported the project to continue that work for 100 priority species coming from the publication of desert tortoise and pollinator habitats, Mojave Desert shrublands. So this is a setup for the stacking software and the photography. Some of these seeds, as you know, are so small, so tiny, and it's important to get the lighting right to emphasize the texture of the seed. I think seeds are so beautiful. They all have their different textures and different features to them, and getting the lighting right was really important to make sure you highlighted those features. My full-time staff member, Catalina, and Lake Mead Plant Nursery, Sondog Plant Nursery was super generous in giving us some space to grow out seedlings. Kelly was super excited because she was like, that's the most species I've ever had growing in the greenhouses at any given time, which was really fun. And we got some really cool results. So these are some of the seeds, just absolutely stunning. I just think seeds are so beautiful. I could look at them all day. And those features, tell a story too about how they move in the landscape, move through the soil column. So it's not just aesthetic, even though I enjoy the aesthetics of the seeds. So we have some phasalias. This is a good example of like some morphological like similarities, but also differences. Very different Manzanilla species. I call this the fried egg and the little brain. For the one on the right, the one on the bottom was actually pulled from one of my gypsum plots. So who knows how long that's been there in the soil column versus a fresh one on the top. Happy Lepidium, Plantago. These are interesting. They kind of get a little gelatinous almost look to them. And of course, the malaria. Gotta love malaria. So here's our seedlings coming up. We took a lot of pictures. I did a lot of drawing. My goal was to have the cotyledons specifically, and then the emergence of the true leaves stage by stage, because it's tough in the field. If you're looking at a carpet of little green things, you're like, these are not all the same little green things, but I don't know what they are. So you either have to come back, watch them and wait, or maybe check out our guide. Here's kind of the final product. So I did 3 illustrations per species and put them together. And I am super excited and proud to say that as of this year, this is published and available for download. There's a QR code if people want to download it right now on their phones. I'll give it a moment. But yeah, it's available online for free for all to use and love on. I've had a couple people tell me they've used it in the field already. So this is for you guys. This is why we do this. It's for those that like to explore, like to put those creative lenses on, look at things a little closer. For the curious, this is for the curious people. So enjoy. So all in all, to close, embrace the duality that exists within yourself. Become the polymath, the Renaissance individuals. Those are individuals that have a broad, deep understanding. They're interested in many different fields and a deep curiosity and passion for exploring different things. You might not be an expert in all those different things, but that doesn't stop you from embracing them. And in many ways, and you'll surprise yourself in the ways that those things can actually blend together and work together. And especially when it comes to teachers, those dealing with younger technicians, don't be that school administrator that told a young person no. Nourish that growth. With that, speaking of nourishing that growth, I always have to give a shout out to my mentors, parents, grandparents, formative professors and advisors through all the schooling and at DRI, and shout out to colleagues at all the different agencies that I work with who I ask for help, and it's just wonderful to have the support of this community. I'll close with a quote. After a certain high level of technical skill is achieved, science and art tend to coalesce in aesthetics, plasticity, and form. The greatest scientists are always artists as well. Thank you.
Good artist.
Right. What do
Yeah. Do we have questions?
Not sure how to stop share, maybe.
Stop share. Yeah.
There we go.
Yeah, we're back up.
Okay. Cool.
All right.
Yeah. Let's see if there are questions.
We have questions for you and Reno. Hang on.
Okay.
Hi.
Hi.
My name is Jackie, and thank you. I grew up in Southern California too. I can totally empathize with the whole La Brea tar pits and all that. Way cool. But when you were drawing the picture of the bear poppy root, what was the composition of the soil that you were going through as you went down that soil column? Was it a hard clay, or was it like crumbly, or what was the texture of it? Like, how was, I mean, obviously it'd have to be somewhat wet for the root to go through it, but what was the overall kind of texture of it? Or was it like layered, or what was it, the soil?
Oh, definitely layered. And actually, I wish Brad was here because he could explain the different soil properties of those different layers more accurately. But there were parts where, I mean, it was almost getting to like caliche layers where it was impossible for us to follow the root. But the root was going, it was finding the smallest ways through those, but we're not able to go any further. But that route could have gone further. Don't know if that answers your question exactly, but yeah, it's actually amazing. And I've done a root excavation on bear poppy one more time, and it was so hard. We got maybe 2 feet in, and it was clear that that root was still going down, but we would have needed a jackhammer to get through. But the root can do it. The plant can do it.
Yeah,
So,
it finds the way.
it finds a way.
Thank you so much.
Yeah.
Christine, if there's any more in here, Tiffany, hang on. No one else?
Yeah. Oh, there's one here in
Okay.
Yeah,
Hey.
at Tule Springs Fossil Beds National Monument, how many species have you documented so far?
Ooh, good question.
Approximately.
Yeah, so we have just over 200 right now. So if we had a water feature in the park, like if Corn Creek was just in the boundary a little bit up that significantly, but over 200 right now. There's plenty more, I'm sure, that I have not found just yet. But that's why I'm like, is it doable as a flora presently. Yes. So who knows? Maybe I will. But yeah, plenty
Yeah.
more to come.
I guess you need different years. of
Exactly.
different precipitation levels.
Exactly. That's why these become lifer projects where you just keep having to go out different times. So
Tiffany,
Speak-
this is Jerry.
lifer projects. Hello.
This is Jerry. Can you
Hi.
hear me?
Yes.
Okay. Have you had a real wet year while you've been doing your flora?
Not yet. Not a really good one.
Okay, because
Yeah.
that would
Not like
sound to
2000,
me like
not
that.
like 2005, like
Yeah.
banner year. Nothing like that. So, although I am going to go out since we had all that late monsoon rain and just poke around and see what's up. So,
Yeah, from what you were just saying, it sounds to me like you need one really super year and then you'd be all set. Okay, thank you.
Thank you. Come on down when it happens and help me.
Have you ever tried making pigments yourself for like coloring things, like stuff in the
Yeah. So I had a question from Marcus asking if I've made pigments. Other than the pigments that the anthotypes come from, I haven't made any pigments lately, but I've been wanting to start doing anthotypes again with some of the pigments from like some of the cacti, like flowers and whatnot. So not opposed, just haven't done it yet.
Yeah, I live near the National Monument and we got 1.5 inches of rain in August. So,
Yeah.
Hopefully that will make some things germinate.
Yep, I need to go check it out. Oh, in fact, we'll be out there next week. Yep. Thank you. Any other questions up north?
When you
Oh, one more down here.
put it on the leaves, what's that? Did you just put light through the image down onto the leaf, or what did you do?
Yeah, so I got a question about the chlorophyll process. So that's where the image was developing on the leaf,
Yes.
right? Yes, so that's actually it's a photographic process. So the sunlight, if you take a film negative or a positive, so that's the reverse of a negative, and you place that over the leaf and then you put it out in the sun, the sunlight is bleaching the leaf chlorophyll through the areas in the image where it's exposed. So the light is actually doing the work, bleaching out the pigment in the leaf. And then you're left with the image.
Bleach out because there was a lack of sunlight in some of those spots that would make, okay.
Exactly, that preserves the color. So, and it bleaches at different rates.
That was fun.
Yeah, it's really pretty. I'll maybe if Marcus has the ability to email to the group. There's a couple artists. There's one artist in particular that he specializes in chlorophyll prints and explores the intersection of just his family's history with like being in Vietnam during the Vietnam War. And his chlorophyll prints on these broadleaf species from that area are just stunning. And he preserves them in resin. They're beautiful. So I'll have Marcus maybe send a link out to his work. Was there one up north?
No, but there's a question online.
Oh.
Leslie DeFalco, did the El Nino signal that hit other parts of the Mojave in 2024 miss Tule Springs?
I didn't. I was able to get out there a little bit, but didn't find anything new, unfortunately. Like nothing where I was like, yes. Although I try and collect just every Gilead that I see on the chance that it might be something new, and then it's not. I'm like, oh, I got the same Gilead again. So yeah, but haven't had like just a really, really good like year. But yeah.
Oh,
That was
is there a no?
Leslie's question. Thank you, Leslie.
Any other questions online? Haven't got any other up here. Thank you so much for your presentation, and thank you for your perseverance to incorporate your art and science and what you're giving us, whether it's in the t-shirt you're wearing and the book that you produced and that you are going to go on producing. Yeah, illustrations sometimes show us something a photograph can't see, so the two together, it's fabulous. And we appreciate you sharing your journey and some of your work. So thank you. And you are our last program. We take a hiatus. There will be no program in December or January. The next program will be in February. Hopefully you'll have some good botanizing down in the south, given that you've had some of these monsoon rains, and your botany season is in the winter, spring, isn't it? Yeah, I was just checking because I haven't been down there very much yet. One thing I did forget, has anyone got any announcements? I forgot to ask for announcements. Sometimes people have things they want to share. No? Well, thank you very much.
Thank you so much. And I hope the guide is helpful if you guys end up using it for different things, and just shoot us an email. We love hearing feedback from folks on if it's helpful. So as I said, it's for you. Enjoy.
Do they have this too?
They do not. People in the South get coloring books. I'm sorry. For those in the North.
From the national.
Yeah. Thank you.
Thank you.
Marcus is coming back up.
Okay.
Get your eye color.